NSF predoctoral fellowship and an NSFD dig, and managed to publish 7 peer-reviewed publications. So he's off to a pretty fast start for a grad student. And I guess he's literally now finishing up and has a postdoc lined up also at UC Davis with a new faculty member there, Andrea Townsend, if I'm correct about that. So in a sense, we get maybe a preview of your finishing talk. You can give us an idea of how it's all turning out, and the grad students here can find out what you guys up at Davis do. I'll leave it at that because the rest of the description here that I was giving really covers, I think, the contents of the talk, and it'd be better if Conor told us about that than I do. Just one quick comment right after the seminar. There's several students that are going to go to lunch with Conor. We've got room for 1 or 2 more if anyone's interested and didn't sign up or didn't get the word. There may also be a little bit of time later this afternoon to meet with Conor individually if anyone would like to later in the afternoon. See me about that afterwards. But anyway, with that, Conor, take it away.
Okay. Thanks, Eileen, for that introduction, and thanks for giving me an opportunity to come talk to you about some of my dissertation work on sexual signaling in common yellowthroats. So one of the things that I find most fascinating about studying animals is the amazing degrees of elaboration and variety in that signaling traits take across a variety of taxa. And so this is certainly true in visual signals like the ones that you're looking at here, but it's also true in other modalities like acoustic signals and chemical signals. And understanding and explaining how these signals has evolved has been a real puzzle for biologists for a long time. So Darwin famously struggled with this. He wrote to Asa Gray in a letter in 1860 that small trifling particulars of structure often make me very uncomfortable. The sight of a feather in a peacock's tail whenever I gaze at it makes me sick. And Darwin was actually sick all the time, so it's not clear how much stock we should put in this letter. But he certainly was really puzzled by explaining how these seemingly costly and seemingly superfluous traits could exist in a world governed by natural selection. And of course, the way that he resolved this was by proposing a special case of natural selection that he called sexual selection, which is essentially competition to gain access to reproductive opportunities. And there were 2 main ways that he thought this could happen. One was intrasexual selection, where members of one sex, like these male bighorn sheep, compete for access to mating opportunities with the opposite sex. And the other is intersexual selection, where members of one sex exert mating preferences for members of the opposite sex, and those preferences are often linked to some aspect of the signaling phenotype displayed by the opposite sex. And so some classic examples here are Mike Ryan's work on Tungara frogs, or Malte Anderson's work on African widow birds. And another thing that Darwin talked about a little bit, and that's really been a major focus of sexual selection research in the last 30 years, is understanding how these sexual signals are actually linked to variation in individual qualities. So why receivers should actually be paying attention to signals at all. So one of my favorite kind of empirical studies of this is work from Michael Jenyon's lab in fiddler crabs, where they've done a series of really elegant experiments using weights added to the fiddler crab claws to show that males that are in better condition can withstand the addition of these weights better. Females prefer males with larger claws, and so by choosing those males, females are indirectly choosing males that are in better condition. And so this is kind of the classic textbook story of sexual selection. It actually took a really long time after Darwin proposed it to work out all these links, but we know pretty well that this actually does happen in nature. But it's really, even though it's kind of the foundational story, it's really a pretty simplistic view of sexual selection. So it's a story of directional selection leading to eventual elaboration of traits over many generations. But the reality, I think, in the last 10 years, as we've gotten more good field data on sexual selection in realistic settings, is that the stories are actually much more complicated. So that's even the case in some of these classic examples, especially like the Tungara frog work. And this is something that behavioral ecologists have been criticized for a little bit, not just studying sexual selection, but in studying any kind of behavior in the wild. Deborah Gordon, in this piece in Animal Behavior a couple of years ago, argued that behavioral ecologists have done a really good job of demonstrating that behavioral traits can contribute to variation in reproductive success, but that it's really time to move beyond studies that just demonstrate or ask why does a trait contribute to reproductive success, and on to studies that ask what are the conditions under which trait X contributes to reproductive success. And I would add to this, when you're thinking about multimodal signals or complex multi-component signals, thinking about what are the conditions under which various components of the signals or different modalities contribute differentially to reproductive success. And I think that this paper makes a nice point. It's certainly something that we should be thinking about. It's a bit of a polemical argument, though. There are plenty of behavioral ecologists that have been thinking about these ideas. So if you scan the table of contents for the last 10 years, just looking at sexual selection, you'll see that there are lots of demonstrations now that sexual selection can vary or be heterogeneous across different situations. So for example, across different sites in the same species, or across different years at the same location, or even on kind of microspatial scales or across different groups within a single location at a single time. But I think it's still the case that this hasn't been really well incorporated into that kind of textbook story of sexual selection. We don't understand that well how this heterogeneity should actually inform our view of the longer-term process of sexual selection. So what I want to do today is talk to you about three kind of case studies from work that I've done on common yellowthroats where we see heterogeneity in the patterns of selection across different contexts or ecological gradients. So I'll give you a little outline of where I'm going. I'll talk about the study system a little bit to orient you, and kind of the foundational work that my collaborators and I have done on yellowthroats over the last 10 years, just filling in those basic relationships between signaling and fitness. And then I'll add a little bit of complexity in thinking about multimodal signaling and how our interpretation of selection changes when we try to incorporate analysis of plumage and song simultaneously. Space use and mating opportunities and the way that habitat and ecological considerations might actually constrain behavioral processes. And then I'll talk about neighborhood fertility and signaling effort and the way that signals might actually be adjusted flexibly to respond to different or changing social conditions. So common yellowthroats are a really common North American migrant. Here in the Bay Area, they're actually resident, but where I work at them is a field site up in upstate New York here, so they're migrant. They're socially monogamous, so this is a picture of one of my field sites in a nice beaver meadow in the southern Adirondacks. In this area, males arrive in early May and defend a single all-purpose territory. Females arrive a few weeks later and settle on one male's territory. And in a typical year, we'll have a few males who are polygynous and attract a second female, and a few males who are unsuccessful at attracting a female. So they'll claim a territory and sing all season, but never actually attract a female to settle there. In terms of sexual signals, there's a few visual signals that might be important for sexual selection. So male yellowthroats have this black melanin-based facial mask that varies quite a lot in size. So you can see this mask is actually about half the size of this mask. They also have a UV to yellow carotenoid-based bib, and again, this varies a lot in size. Again, this bib is about half the size of this large bib male, and that's pretty typical of the variation that we see at our field site. The bib also varies a lot in color, so both the hue of the bib and the brightness. And we can quantify that by plucking feathers from the center of the bib and using a reflectance spectrophotometer to generate these reflectance spectra. And so what you're looking at here is a peak in the UV range, which we can't see, but the birds can, followed by a dip, and then another peak in the yellow range, which is mostly what we're seeing when we look at these birds. And a little bit later on in the talk, I'll mention three different color metrics that are derived from this spectra, so I just want to mention them now because it's easier to see them when you're looking at this. So UV brightness is essentially the high point of this UV peak. Yellow brightness is the height of this yellow peak. And carotenoid chroma is a ratio that takes the height of this yellow peak and the low point of this trough. And that's actually a measure of spectral purity that's related to the amount of carotenoids that are deposited into feathers during feather growth. So each field season involves a lot of basic census work, so I catch all the adult males and females, put color bands on, you can see here, for visual identification, take morphological measurements like wing and tarsus length, and take blood samples from all the adults and all the nestlings so we can do microsatellite-based paternity analysis. But the vast majority of the field season is actually spent finding nests. So yellowthroats are ground nesting swamp birds in dense swampy habitat. And this bottom center picture here gives you a pretty good idea of what a yellowthroat nest looks like from about a foot away. They're really sneaky when they're building these nests, and it's actually an incredible amount of man hours that have gone into finding these nests, both my own and a lot of field assistants over the years. And the result of all that effort has been a pretty good database that we can start to ask some sophisticated questions about sexual signaling in. So we have 8 years of breeding data at these two field sites. We've watched about 200 males and about 150 females over those 8 years, and many of those individuals survive and breed in multiple years, so they return to almost the exact same territory each year. So we have 335 observed male breeding seasons and 232 observed female breeding seasons. And all of those man-hours of nest searching resulted in 486 nests and produced 808 nestlings that we actually sampled. So these are nestlings that survived to day 5 and we were able to take blood samples from. There are many more nestlings that hatched or eggs that were laid that didn't survive until day 5. And one nice feature of these study sites is that they're both swamps that are surrounded on all sides by dry woodland habitat where there's no yellowthroat territories. So we don't get any intruding males coming in from areas that we're not monitoring. So we're able actually to assign 100% paternity to all the nestlings that we sample in the site, and we're pretty confident that the males in our site aren't siring nestlings in adjacent areas. So there's not really anywhere for them to go. And it turns out that of those 800 nestlings, about 18% are extra-pair young, so we can actually assign for each male in the population both the within-pair and extra-pair reproductive success in each season. Okay, this is going to be a whirlwind tour of what we know about sexual selection in yellowthroats. So this slide could probably be another whole hour-long talk, but I just want to give you the foundational basic story of sexual selection. So my collaborators and I over the last 10 years at this site have shown that different aspects of plumage size and coloration related to the bib or mask are related to a lot of different things that might be important for fitness, variation in fitness. So for example, male dominance and female preferences in aviary trials, both within and extra-pair mating success in the wild. A lot of things that you might think are related to male condition or male quality. So some measures of immunity, male body size and provisioning rates to nestlings, oxidative stress measured as DNA damage to red blood cells, survivorship across years and circulating testosterone levels, and some things about the offspring, like offspring sex ratio and condition. And then the last thing that I want to say here is that I'm not going to talk much about this in the rest of the talk, but there are also these pervasive relationships between the patterns of selection that we see, the condition dependence of ornamentation, and male age and experience, where we actually see what looks like different patterns of selection on different age classes that change in relation to the way that condition dependence of ornaments changes. And that heterogeneity between age classes is kind of what, back at the beginning of my dissertation, got me thinking about the way that we might be seeing heterogeneous selection within this population and led to the studies that I'm going to talk about now. Okay, so the first thing that I want to go on to is thinking about multimodal sexual signaling. And you'll notice that on that last slide, I didn't say anything about song, but of course yellowthroats have a song too. And turn the volume up. I'll play it for you here. So it's a witchety, witchety, witchety is the mnemonic. And so I was interested in adding to our analyses of visual ornamentation by looking at song variation and whether there might be sexual selection for song performance. But yellowthroats have a song, so they have a single song type that they repeat all season. They don't have a lot of the features that people have typically looked for when they study sexual selection in songbirds. So they don't have a repertoire. You can't look for variation in repertoire size. Because they don't have a repertoire, they can't match songs with their neighbors because they don't change their song type. And they also don't have trilled components to their songs like a lot of sparrows have. So you can't look at the same kind of fine motor performance constraints on song production that seem to be important in sparrows. But what I became really interested in is when you have this complex behavior, whether the variability in producing their own song might actually become an informative signal. So birdsong, especially songs like this that have a lot of frequency modulation, is a really complicated behavior. It involves coordination of musculature, the brain, and vocal tract shape, so the posture that the bird sits in can actually change the song production quite a bit. And because of this, it might be really difficult to recreate the same exact song perfectly from one rendition to the next, which is essentially what yellowthroats seem to be doing. And this might be especially true as birds fatigue, so when they're doing a song bout where they're singing at a really high rate for a sustained period of time, and they can't switch to a different song type. So in this situation, individual quality might actually be signaled by low variability within a bout in song production. And you can imagine this from a receiver perspective. So if you imagine two females listening to two different males, this female on the left is listening to a male who sings his song a little bit differently every time, and this female on the right is listening to a male who sings the exact same version of his song every single time. And you can imagine that these could be quite different sensory experiences for a receiver. So I'm not going to go into the methods that I used here, but I'll say briefly that I took vocal recordings with a shotgun microphone from each male in the population and used spectral cross-correlation to look at the consistency of producing that song for each male across a bout. And I'm happy to talk about that more later, but I'm just going to go into results here. So the first thing that I found is that males that sired at least one extra pair young, so males that were successful extra pair sires, were much more consistent singers. So it seems like there is some evidence that females are actually paying attention to this song consistency within a bout. But the question that I started this section off with was thinking about how song and plumage are actually interacting with each other. So to get at that, what I did is go back to some previous work we had done on plumage ornamentation and add this song consistency measure to those same multivariate models. And what you see is, actually, I think, really interesting. So when you look at within-pair mating success, so whether or not males were able to attract a social female to their own territory, it turns out that mask and bib size are really important. So males that have larger masks and larger bibs are much more successful at attracting a female to settle on their territory. But when you look at extra pair mating success, you see a really different pattern. So these visual ornaments don't seem to be important at all, and there's no relationship there. But what you do see is that this song consistency relationship that I showed you on the last slide holds up. So even after controlling for this variation in visual ornamentation, males that are more consistent singers are more likely to sire extra pair young. So I think the main result here is that whole song consistency is related to reproductive success, and I didn't show this, but there's also some relationships with survival, so it seems like it might be indicating male quality. And when you actually consider song and plumage traits simultaneously, only song consistency predicts extra pair mating success, and only plumage ornamentation predicts within pair mating success. So I think this is a good example of a case where multimodal signals are actually functioning in different contexts, and the contribution to overall fitness is going to depend on the specific conditions. So what I mean by that is that if you have a population where within pair reproductive success is really important, you might expect to see stronger sexual selection on ornamentation. And conversely, if you have a population where extra pair success is very important, you might expect to see strong selection on song consistency. And the last thing I want to say about these song recordings before moving on is just to point out that there's also variation within males. So all the results that I showed you so far were restricted to recordings that I made early in the season when males were unmated. But I also have a lot of males where I have another recording later in the season after they have fledglings or an incubating female in their territory. And it turns out if you connect those recordings, so each line here is connecting one male recorded early and then late in the season, almost all of these males decline in consistency quite a lot across these two different contexts, so also across the breeding season. And this difference within males across the season is actually bigger than the magnitude of difference you see between successful and unsuccessful males in those early recordings. So even though we see apparently strong selection on song consistency, we also see this amazing degree of flexibility in signal production. And I'm going to come back to that in the third part, but I just want to point it out here. Okay, so I want to move on to thinking about space as a constraint on behavior. And I think that as field biologists, it's really easy to go out and collect all our data, bring it back to the lab, and get it into a spreadsheet. And then when we start analyzing that data, to forget that animals actually exist in the real world. They're not numbers in a spreadsheet. And when we think about sexual selection in particular, we often analyze our data as if individuals were free to choose from and display to every individual in a population. But of course we know that's not true, right? They're constrained by all kinds of real-world things. So one really important constraint might be that space use can actually structure contact and opportunities to display to different individuals in a population. And this is particularly true for territorial songbirds, where the spatial distribution of territories and nests varies at both local and landscape scales, depending on abiotic or ecological factors. So yellowthroats are only present in swamps, so the distribution of that swamp actually sets up where the territories lie. So what I wanted to do here was first of all ask whether extra pair paternity is spatially limited. So do we see any evidence that there are constraints on how far males are able to travel in order to sire extra pair young in nearby territories? And if we do see evidence of a constraint, does a spatially explicit model that actually incorporates information on that variation in opportunities to acquire extra pair mates explain more of the patterns of overall variation in extra pair mating success, and increase the understanding that we have of the relationships between plumage ornaments and extra pair mating success? So the way that I did this is go back to that big database, and for every male, or for every nestling that we had that was an extra pair young, I calculated the distance from the nest that it was born in to the center of the territory where the true genetic sire was, and I'm showing you just a histogram of those distances here. And the thing that jumps out is that 70% of extra pair young are sired by males that are only 200 meters or less away. So this is really close. If you're in a territory, you can hear a male singing 200 meters away. So this is a really short distance for extra pair young to be sired. and 97% are sired by males within only 400 meters. So even at the outer limit, it's still a pretty close distance. And even these 3% that you see out there are a couple of weird cases where one year I had a male who was polyterritorial, and one year I had a male who settled on a territory and then moved partway through the season. So it really does seem like there are these strong spatial constraints on how far we should actually consider nestlings being potential extra-pair reproductive opportunities. And I'll show you two males just to illustrate this a little bit more. So these are the center territory here in black is showing a focal male. The inner ring is a 200-meter radius, and the outer ring is a 400-meter radius. And you can see that this male on the left has a single neighbor within 400 meters. And we sampled 4, it's a little hard to see the number, but we sampled 4 nestlings from that male's territory. Whereas this male on the right has about 10 neighbors, and we sampled something like 25 nestlings from those territories. So you can imagine that this male on the left might have the most attractive plumage ornaments and the most consistent song, but there are just so few opportunities to sire extra-pair young that he might actually not be that successful. And this male on the right might have a pretty typical signaling phenotype, but he has so many chances to sire extra-pair young that he might actually end up being pretty successful. And you can quantify this a little bit more formally by calculating the opportunity for selection across different breeding densities. And so this is a metric that parses variation in total reproductive success into different fitness components. And so here I've divided variation in reproductive success into within-pair and extra-pair components. And you can see, starting at the lowest density, that almost all of the variation is explained by within-pair success. And extra-pair success explains almost nothing. And so what that's telling you is not necessarily that sexual selection is happening, but it does give you an upper bound estimate on the strength that sexual selection could take. And so at these low densities, there's so little variation in extra-pair mating success, essentially because nobody's really siring extra-pair young, that there's no way that sexual selection can operate, regardless of how strong female preferences might be or how informative male signals might be. As you move up to medium densities, you start to see more contribution from extra-pair mating success, and as you move up to the highest densities, you see actually an almost equal contribution from within an extra-pair mating success. So what I wanted to actually do is see whether we actually see variation in selection across these densities. And so to do that, I went back to that same kind of multivariate framework that I showed you in the first section. So again, this model includes the plumage ornaments that I talked about earlier, the 3 bib color measures, UV brightness, yellow brightness, and carotenoid chroma, and then bib and mask size. But here I've also added information on breeding density. So this is essentially a count of the number of nestlings within 400 meters that a male could have sired, and then two-way interactions between that density measure and each of those ornament measures. And what you see when you fit this is that there are some interactions that pop out. So I don't want you to look at this whole table, but just focus on these interactions. So what this is essentially saying is that the relationship, the predicted relationship between all 3 of these bib color measures and the likelihood of siring extra-pair young changes across different densities. And rather than interpret these model coefficients, I'm going to plot for you what this is saying. So I'll orient you in this first plot here. So this is UV brightness at low density. The x-axis for all of these will be standard deviations above or below the population mean. And the y-axis is the model predicted likelihood of siring at least 1 extra-pair young. And even though it looks like there's a positive relationship here, this is the maximum likelihood estimate from the model, the confidence interval shown in the almost invisible gray lines here is actually really wide and crosses 0. So there's no evidence that there's any selection going on here, which is what we would expect, right? Because there's no variation at these low densities. You see the same thing for the other 2 color measures, but as you move up to higher densities, you start to see some patterns emerging, and when you move to the highest densities, you see really strong, consistent selection favoring males, so a positive relationship between the likelihood of siring extra-pair young and yellow brightness, and a negative relationship for UV brightness and carotenoid chroma. So I think the kind of take-home message here is that space actually matters. So when you actually consider spatial data and add that to the model, you can explain a lot more variation and actually reveal new relationships. So if you remember in the first talk, I said that we initially didn't think that plumage was related to the likelihood of siring extra-pair young at all. And it's not at a big pooled population level, but when you actually start to consider the variation in opportunities to mate, you do see these patterns emerging, at least at the high density areas. And I think another really interesting point to take away from this is that there's no evidence here that, or at least I haven't shown evidence that the behaviors actually change across densities, but the behavioral outcomes are actually shaped by ecological contingencies. And so what I mean by that is you could have the exact same female preferences and the exact same honesty of male ornaments, but because of the way that space actually structures interaction between those individuals, you can get different behavioral outcomes. And so if you want to actually understand or predict evolutionary outcomes or understand the evolutionary history of signals, I think it's going to depend on understanding both how selection varies across different ecological gradients, like density, but you can imagine other gradients here, and also the relative percentage of breeding population that experiences each population. So for example, if you have most of a population at high density and just a few individuals at low density, that might be a different dynamic than a population where individuals are spread across low and high density areas. And I think this is potentially a real problem for behavioral ecologists because, as Judy Stamps pointed out in this animal behavior paper a couple years ago, we're often choosing our study sites specifically to get large sample sizes and potentially biasing them towards areas where there's higher than typical breeding densities. And it's not really clear, as I've illustrated here, when we understand processes at high densities, how well that understanding will translate to other breeding densities or other ecological gradients. Okay, so in my last section, I want to combine what I've talked about in the first two in thinking about space, social context, and song production. And so I hope I convinced you in the last section that space matters and actually sets up behavioral interactions between individuals. And I also alluded at the end of the first section to this idea that social context might matter and that males might actually be flexibly adjusting their signal effort or signal allocation across different social contexts. So I want you to think about a male yellowthroat arriving on the breeding territory in early May. We typically think about what's going on in their own territory, right? So they arrive, they might attract a female, they go through nesting, they might have a predation and renest, and eventually they produce fledglings, and that's the end of the season. But the reality is that that whole time there's also this complex social environment going on neighboring territories. And we know that extra pair fitness is important, so the whole time that the male is taking care of business on his own territory, there's also females that are arriving on neighboring territories. They become fertile. We have a lot of predation, so females build a new nest in the middle of the season, and you get a lot of variation in social context going on throughout the season. So what I wanted to know is, first of all, do males seem to be responding or monitoring these changes in social context in neighboring territories? And how should signaling effort actually be allocated to adjust to this complex social environment? And do males actually adjust their singing output to respond to this? So to tackle this question, I got these automatic recording units, which are these great devices that can be placed out on trees in the middle of males' territory. They're waterproof. They can be set to record on a fixed schedule, so you can get a lot of recordings throughout a day. And you can get a bunch of them and leave them out for a long period of time, rotate them through males' territories, so you can actually get observations, detailed observations of individual males across many days and of many males across the breeding season. And before I go into some data, I just want to set the stage here for you. So this is a good time in the talk to take a nap if you're feeling a little tired. You can wake back up for the punchline. But if you were in the southern Adirondacks at this really pretty field site on a pristine beaver meadow in early May, it would be a little bit darker than this. But the first thing that you hear before dawn is the spring peepers. And even before the spring peepers are done, you would hear the first yellowthroat coming in. So they're really prolific singers, especially in these habitats. Most of the songs that you hear in these recordings are yellowthroats, so there's the first yellowthroat. And then you hear some other birds, like a gray catbird. These are one of the most annoying ones in the recordings, because they'll sing for half an hour straight. And then Veery sings here from the edge of the swamp. And we also get a lot of other swamp specialists, like a song sparrow that comes in here. And then you get the yellowthroat coming back in, and now he's joined by another yellowthroat, so they're setting up territory boundaries, counter-singing back and forth. And then in this recording, they're actually joined by a third individual singing on the next territory. And so in this one recording, you can hear all three of these individuals singing across their territorial boundaries. And at the end of the recording, one male intrudes on the focal male's territory, and you hear a rattle and chip. So these are aggressive calls where the focal male is probably chasing that individual back out of the territory. And so I think it's pretty clear from this recording that these automatic recording devices can really easily generate terabytes of data. And they also can make some great kind of natural sound recordings that you can listen to when you're going to bed. So if you're interested, see me after the talk, I can give you some of those. But what can they actually do to answer interesting science questions? I think they can actually do a lot. So what I've been doing working through these sound files is using a semi-automated song detection algorithm. So what you do for this is feed the computer a training set of known yellowthroat songs and ask it to go through all of these files and pick out everything that it thinks is a yellowthroat song. It does an okay job most of the time, but I say semi-automated because you do have to go through and hand check all of these. So you'll see that it picks out Yellowthroat songs for the first few here, and then the last one is something that doesn't look anything like a Yellowthroat song. So you have to go through and throw those out by hand. But in the end, you can get a big ID'd data set of Yellowthroat songs. The question you might be wondering is, how do you know which individuals are singing those songs? And this is where Yellowthroat breeding biology and song biology actually becomes really convenient. So they have small territories. So when you place one of these recording devices in the middle of a male's territory, you can be pretty confident that you're getting recordings of all of their songs. They don't really sing beyond the boundaries of their territories. And because they don't really sing beyond, you're only really getting recordings of the focal male and 2 or 3, at the most, neighboring males. So you have a limited sample, a limited number of males that you're actually trying to ID songs out of to begin with. And then the other nice feature of Yellowthroats is, like I said earlier, they don't have repertoires. They have a single song that they produce. And even though this song is really consistent within males, there's variation between males. So I'll show you that here. So these are 3 songs taken from different recordings of the same male. And you can see that just looking at the spectrogram, they're almost visually identical. This is true even if you look at recordings that were taken years apart from the same individual. Once they're adults, they pretty much don't change their vocal signature. But when you look at other males, and these are just 4 random males that I pulled out, you can see that there's actually a lot of variation in the structure of the songs. So by combining that known location of the recording unit and these known ID reference samples, you can actually do a really good job of picking out every song produced by a focal male in each one of these recordings. And so following that procedure, in 2 years of recording, I've built up about 1,500 hours of recording, so these are spaced out throughout the day. A little over 1,000 days of observation from 39 males, and so this turns out to be about 27 days per male. So it's about a 75 to 80 day breeding season, so 27 days of observation spaced out throughout that season is actually a really good representation of how song output is varying across that entire breeding season and across changing social context in each male. And so I've ID'd about 125,000 songs produced by focal males, so that's not including ones that were thrown out by neighboring males. And I actually have another year of data that I'm finishing up, and hopefully will be the last time I ever do this right now. So this number is actually pushing closer to 200,000 songs at the moment. So it's a pretty rich database that we can then go ask how males are actually changing or flexibly adjusting their song production, total song output across different contexts. So the first thing I want to talk about a little bit is what goes on in the home territory. And I'll tell you a story of this one male. So this is PR.W. So he has a purple and red band on his left leg and a white and USGS band on his right leg. He arrived in 2011, and early in the season, on May 18th, sang a ton of songs. So he's singing more than 10 songs every 2.5 minutes in the morning, and even later in the day, he's singing more than 5 songs every 2.5 minutes. Just 10 days later, he attracts a female, and his song production drops off drastically. So this song profile represents about 2,000 songs. So even though these songs are pretty short, 2,000 songs for a 10-gram bird in a single day is a lot of time singing. Here, he's dropped off where he still has a peak in the morning, but his total song production is down in the couple hundred range, only 10 days later. A little bit later, that female is laying eggs. And you see on this day, when she was laying eggs, he actually did a high-rate dawn chorus bout for a very short time, and then dropped off for the rest of the day almost to 0. And then the reason that I picked this male is that his female was actually depredated partway through the season. And so in July, when most of the males are kind of winding down their breeding efforts, he actually is unmated again and goes back up almost to these early season levels. So he still has this dip, which I'm not sure how to explain, but his total song production is above 50% of, or about 50% of what it was when he was unmated early in the season. And you can look at this a little bit more quantitatively, pooling across males. So what I'm showing you here is the averages for dawn chorus and for daytime song, when males were either unmated, had a fertile female on their territory, or had a non-fertile female, which means the female was either incubating or they had fledglings on the territory. And for dawn chorus, males sing the most, unsurprisingly, when they're unmated. They drop off pretty drastically, but still sing a fair amount when they have a fertile female. And then they actually rebound and go back up once that female is incubating or they have fledglings. For daytime chorus, daytime song, you see a little bit of a different pattern where males sing a ton when they're unmated, and then that drops off when they attract a female and really never goes back up again. It's pretty low for the rest of the season. And just because I have these data, I'll show you this in a little bit of a different way. I think it's one of the things that these automatic recording units can do really nicely is generate these curves that show you song production over the whole day. So here what you're looking at is actually the recording schedule that I use. So you can see there's a dawn chorus recording that's a long block, and then spaced out recording throughout the rest of the day. The blue profile is the average unmated male, so they have a really high rate dawn chorus, and then they actually sustain a pretty high rate all throughout the rest of the day. Once they attract a female, they go to this green fertile stage, where they do go up to a nice peak at the same time during dawn chorus, but the maximum rate is only about half what it was when they were unmated. And then it drops off really precipitously and stays low through the rest of the day. Once that female is incubating, they again have the nice dawn chorus peak, and it actually reaches the same maximum rate that they reach when they're unmated. So they're going up to a really high rate during that dawn chorus, but then again it drops off and looks just like the fertile stage. So it's interesting that you see these differences across these different times of day. But the real question that I started off with was, do we actually see adjustment of song production to what's going on in neighboring territories? So are males paying attention to fertility of neighboring females, and are they actually changing their own signaling in response to that? And so what I did to get at this is, for every female in the population, I defined a fertility window, which started 5 days before they laid their first egg in the nest, and went to the day that they laid their penultimate egg. And on every day that I had recordings for a male, I counted the number of fertile females that were within 400 meters of his territory, excluding his own social mate. So remember, 400 meters is the distance where there's at least a possibility that they might be engaging in extra-pair copulation on those territories. And a couple features of yellowthroat biology that were incredibly frustrating in previous seasons ended up being really nice for this study. So they have very frequent predation. Remember, the nests are really hard to find, and over 60% of them end up getting depredated. Fortunately, what yellowthroats do when they have predations is that they rebuild. So we've had individual females that will rebuild up to 6 nests in a season. So this is something like 15 to 20 eggs laid in a season for a 10-gram female. It's pretty amazing. But what this does for this study is it actually sets up this quasi-experimental variation in the fertility status of neighboring territories. And what I mean by that is I wasn't actually going out and manipulating fertility status on neighboring territories, but with respect to a focal male, you can think of predation events on those neighboring territories as being essentially stochastic. And because females renest all season, you get this variation in neighborhood fertility status that's decoupled from date in the season and decoupled from the social status on a male's own territory. So you can actually piece apart statistically whether males are responding to that change in fertility. And so I did this using a model selection approach. So there are a couple of candidate models that I fit here. First is a null model, just predicting that song production is not explained by anything that I measured. Next is a couple social-only models. So these include information on what's going on with a social mate, so whether a male is unmated, has a fertile female, or a post-fertile female. And then also some abiotic factors like temperature and date in the season. And then I fit a series of what I thought of as more interesting models. So these include information on neighborhood fertility status. So again, these all include the social information because we know that there are drastic responses to what's going on a male's own territory and his pairing status. But what I wanted to know is, after you actually control for those, do you see males also responding to changes in fertility status on neighboring territories? And again, some of these include these abiotic factors like temperature and date that might be important for song production. And so what you see when you fit these models is that the vast majority of the weight gets thrown onto models that include the number of fertile females. So including that in the models makes for a better model fit. This is showing just dawn chorus song, but daytime songs are very similar. The table looks almost exactly the same. The order of models is the same. The weights are a little bit different. But rather than show you all the coefficients from these fit models, I just want to show you what the models overall predict for the relationship between singing and neighborhood fertility at dawn and during daytime. And so that's what this is showing here. So the black lines are model predicted fits. The blue dots are a subsample of some real data, just to give you an idea of what this looks like. So the left plot here is showing dawn chorus, and the right plot is daytime songs. And on the x-axis, you have the number of fertile females in 400 meters. And what you see is that for each additional female within 400 meters, after you account for what's going on with the male's social mate, they actually are predicted to increase their singing production by about 10%, which might not seem like a huge amount, but when you consider that some of these males might be singing 2,000 songs in a day, and they might have 3, 4, or 5 females that are fertile around them on any given day, this actually can add up to a pretty substantial increase in song production. And pretty amazing that these males are actually doing this not just in response to any female being fertile, but more females fertile in the area seems to lead to a larger response. I think that one question that kind of derives directly from this is how do males actually know about neighborhood fertility? So it seems pretty clear that they're responding to what's going on, but how are they actually monitoring and acquiring information about neighborhood fertility? And I think one possibility is that they're actually actively visiting neighboring territories and checking on fertility. There's some evidence in another population of yellowthroats in Wisconsin that supports this, where radio tracking studies have shown that males that foray off territory more are more likely to sire extra pair young. Although it's not clear whether they're foraying to acquire information or whether they already know that those females are fertile and are then visiting them. One possibility that I think, given the data that I've shown here, is really interesting, and I don't have data to support this, but I call it the curious incident of the bird in the nighttime. So you probably all know the Sherlock Holmes story where he's able to solve a murder because the dog in the night didn't bark, and therefore the murderer must have been someone who was familiar with the dog. I think when you have this kind of tonic communication system where birds are just singing all the time, you can actually have the absence of a signal be informative. So males are just so responsive to what's going on with their own female that they might actually be inadvertently signaling to rival males that their female is fertile. And presumably this is something that males just have to do, right? So if you watch males in the field, when a female arrives, they follow her really closely, they're trying to copulate and mate guard, and they presumably just can't sing. But by doing that, they're telling all of their rivals exactly what they don't want their rivals to know, right? My female is fertile, you should increase your song and try to mate with her. So I think that the main result here is, so one interesting result here is that we actually can quantify with these automatic recording units that males adjust their song output in response to changes in their social territory. And this is something that ornithologists know from being in the field, but I think it's actually something that's been hard to quantify in a really good way for a large number of individuals, because you would just need an army of field assistants out counting the song production on a lot of different territories every day throughout the breeding season. And the really interesting thing is that after you control for that, males also seem to respond to changes in the potential for extra pair reproductive opportunities. So the number of fertile females in the area affects how much they're actually singing. And the fact that they're changing their song production in response to neighborhood fertility suggests that males are engaging in some kind of sophisticated surveillance of neighboring females up to 400 meters away. I don't know exactly how they're accomplishing that surveillance, but I think one possibility is that they're actually cueing in on the song production of rival males as a way to gain information on neighborhood fertility. So just to wrap things up and tie these three sections together, I think, as I've demonstrated here, that heterogeneity in sexual selection across a lot of different factors is coming to be seen as the norm rather than the exception. So I think everywhere that we're looking for it, we're starting to see it. And given that that's the case, if we want to understand and explain the evolution of sexual signaling, we need to incorporate an understanding of how changing contexts change patterns of sexual selection, whether those be social contexts or ecological gradients or spatial contexts. And I'll just point out a few areas where I think this has implications. So on a kind of microevolutionary scale, there's a clear implication in understanding or reconciling seemingly contradictory results between different study sites or different years at a single study site when there might be an ecological gradient that we haven't accounted for, explaining those differences. I think there's also a role for understanding the relative importance and the interactions between signals in different modalities or different components of signals and under what circumstances those different components are most important. And on a more macroevolutionary scale, one kind of obvious implication is thinking about the maintenance of genetic variation in signaling traits. And this is something that people have written about in a few papers pointing out that heterogeneity might contribute to resolving the Leck paradox. I also think there are potentially interesting avenues of research to think about the patterns of signal evolution across different clades and the degrees of heterogeneity that those clades might experience in terms of the habitat they're living in, how that might actually contribute to the types of signals or the degrees of elaboration that you see across different clades. Unfortunately, quantifying heterogeneity in sexual selection is challenging. It's a lot harder than just doing two-choice trials in an aviary. I think that those trials are great. They demonstrate a lot of proof of concept that female preferences exist, but they don't really tell you how sexual selection actually functions in ecologically realistic settings. And if you want to get to that, I think, and I'm a bit biased here, but that the way to really get there is long-term studies with known individuals where we can actually track fates of individuals and look at selection across different contexts. and particularly studies that look at animals at multiple study sites, multiple years, over a long time. This research was done by a lot of people, not just myself, so I have a ton of field and lab assistants to thank for all their contributions, some funding sources that paid for different parts of it, and a lot of collaborators and advisors who contributed to all the work that I talked about today. And with that, I'll take some questions.
The lights back there. So I'm going to let folks who have a 1 o'clock obligation scoot out quickly. Take a few questions. Okay, go for it. Liz. Katie.
So you first showed results that the color characteristics of the birds didn't affect extra-pair paternity mating success, but you then showed that an interaction with them did influence the extra-pair mating success. Do you think the first results where it didn't influence it was just because you weren't yet testing the interaction?
Yeah, so the interaction with breeding density or with the opportunities. Yeah, I mean, I think it is because that first study was a smaller sample size. That the breeding density has 6 years of data, and the other one had 3 years of data. But I really do think that there's something going on across those densities where at a pooled population level, you have individuals with lots of different opportunities. So it's essentially like adding noise. And when you actually control for that by thinking about the opportunities that they have, you see those patterns emerging. It's not clear that you'd always want to control for those, right? Because I mean, that's really what's going on at a population level. At the broad population level, there aren't really those connections. It's only when you control for those opportunities. But I mean, that's kind of a statistical technique to control for those. If all of the individuals in the population were at high densities, then you would expect to see those patterns emerging. And yellowthroats, I've thought about trying to do kind of a quantification of where the populations are breeding, but it's not clear to me what percentage of the population is in high densities, because you have these really dense breeding aggregations in bogs, even a lot bigger than the one that we work in. And then you also have a ton of yellowthroats that are like in an individual territory in a drainage ditch on the side of the road. And there are a lot that are breeding kind of individually like that, and presumably they have, presumably a lot of them are not attracting females to those territories, but the ones that are have no opportunity to sire extra pair young. So it's not quite clear how that would impact the population level trajectory of signal evolution.
So, in thinking about the population trajectory for signal evolution, then too, it matters sort of which habitats are causing the greatest amount of recruitment for the next generation.
Yeah.
And I was wondering if you had a sense in terms of both overall and per capita. I mean, I would assume that the high density may be better and is contributing more individuals, but I don't know if on a per capita basis.
I mean, I don't.
Do you have a sense about how that works out?
I don't see there are no clear differences in total fledgling production across these different densities. So you can imagine kind of scenarios where it might be better for an individual to be at high or low density, right? Because at low density, you don't really have a threat of losing paternity in your own nest. You're kind of giving up the option of gaining extra pair paternity in other nests, but extra pair mating success is really age dependent too. So might be this trade-off. This is something that I actually really wanted to find when I started looking at that breeding data. There might be a trade-off where as a young male, there's almost no chance of siring extra pair young. So all you can really do by being in high density is lose. You can lose paternity in your own nest. But as an older male, once you have more attractive signals, you have a potential to sire extra pair young. But the males don't move much between years. So I really wanted to see whether individuals were making movement decisions based on their own potential reproductive success. And there's some indication that young, second year to after second year birds are more likely to move. Most of them don't move at all, but it's not a strong enough result to really say that there's a pattern there.
And just to clarify too, you had mentioned that overall there's maybe not a difference in number of fledglings for males in low and high density on average, but could you, I guess, just curious, so you don't have an opportunity for extra pair population of the males in your low density. If you're in high density, the males that are getting lots of extra pair population, so they also tend to have less loss of population, or is it essentially everyone?
Yeah,
is it kind of just everyone's messing around?
The only thing that I've ever found that was related to your likelihood of being cuckolded is how many fledglings you have. Where if you have more, you're more likely to be cuckolded, but it doesn't seem to relate to any of the things that are important for extra pair fitness. It doesn't seem to relate to any of the signals or breeding densities. Yeah.
One more, yeah. And
This talk made me think about how quantifying the amount of sexual selection in the population and sort of like how much mate choice is actually going on. So I was wondering, do you have any sense about if you had your most simple model was just like one color trait and extra pair paternity versus your most complex model, how much more variation is explained by that?
It's the color things are a little bit complicated because a lot of those color measures are correlated in ways where you kind of need to include, like to describe, even though it's three, even though you're putting more things in the model, you actually need them all to explain the color more fully. I'm not sure.
So if you add like density to that.
Yeah, I mean, adding density explains a lot more of the variation for extra pair mating. I don't know off the top of my head what the number is, but yeah.
I guess, but it explains extra pair mating, but not necessarily reproductive success, right? So then, sexual selection isn't just about extra pair mating; it's also about who they choose to be their social mate.
Yeah.
Right. So you need to be careful about talking about sexual selection as extra pair mating.
Right.
It's not.
Right. I mean, the reason that we talk about sexual selection as extra pair mating in this system is that, at least for the older males, there's very little variation in within pair mating. So they mostly, I mean, we don't know anything about offspring quality. So potentially there might be really important things going on with the quality of offspring produced by different mates. But the number that they're producing is essentially, they basically all fledge one nest. And the nest has 3 or 4 nestlings in it. So there's not a lot of variation in within pair mating success. The way to really increase your seasonal fitness is by getting extra pair matings. Some males, a few males have like 10 extra pair young. So they can actually double or triple the typical seasonal reproductive success.
I think kind of partly because we have to clear this room because the curators have to meet with the external IB review committee in 10 minutes. But thank Conor, and feel free to grab him either a couple want to go to lunch or I can go and meet with them later today. But thanks, Conor. So Katie, you're going to lunch, sure. It's you, Jeremy, Aaron, and
Yeah.
I wasn't sure I wanted to ask it from the whole group. I'm not really but don't the fairy wrens actually have females actually visiting other male territories at night when most of them are
Yeah, and we haven't done any radio contact on females. I would love to know more. The thing is, I would love to know a lot more about the female behavior. They're just so hard to observe. So you need to put radio tags on. We usually don't catch them until they're already nesting. And they kind of arrive one morning and they're on a territory and they've chosen that mate. So I would love to actually know like the choice process that goes in when they're visiting other territories. The fairy wrens are nice because they're not migratory, so they can actually work at those field sites year-round and get individuals where they're putting things on early and then watching them going into the breeding season. And they can look at pre-breeding season events and how those relate to what happens in the breeding season. We can't do that easily. Yeah. But yeah, it's
amazing that you can actually find all these birds in that kind of habitat.
yeah, it's a lot of
So
time.
you're walking in water for months on end.
Yeah, it's actually like grassy. We're not in the really thick swamp, so it's pretty passable. But a lot of the swamps where there's more birds, we don't work in because we just wouldn't be able to.
Yeah.
So it shows one it's kind of actually probably a little bit marginal habitat. It's not where the biggest aggregations of birds are.
Uh-huh.
But the ones where they are just completely, and in upstate New York they're all full of Phragmites, which is this reed that's this tall. It's kind of like tulle.
It would be impossible to see.
You wouldn't be able to, you wouldn't have to see them.
So I assume that based on your results, that birds must be avoiding the edge. I mean, so you've got this edge habitat, right, where good habitat meets bad habitat, which the birds aren't really crossing. Seems like that would really reduce your opportunities for.
Yeah, I mean, they're actually, so it's kind of all edge. It's a beaver meadow that's on an old stream. So there's a string of territories where mostly they have one neighbor like north and south, and then the east and west is woods. So they're
Yeah,
almost
that would
all
be
on the edge.
interesting.
It's not really like
Got
There's
it.
some other
It's not a
boulders that are
circular
edged.
habitat,
Yes.
it's like a long.
But there are circular habitats. I mean, there's other bogs that
No,
are
not that button.
circular.